بسم الله الرحمن الرحيم. النهارده هناخد اول محاضرة في الجينيتكس. الاوتلاين بتاعنا جينيتكس، genes, genome, the difference between genetics, genome, structure of DNA replication, types of RNA, finally protein synthesis. Firstly, what is the difference between genetics, molecular genetics, gene and genome? Genetics is the science of heredity and variation in living organisms. Molecular genetics is the study of agents that pass the information from generation to other. Genes are long polymer of deoxyribonucleic acid or DNA molecules. Genome divided into two parts, gene and ome, meaning all genes. The DNA structure formed from building block of purine and pyrimidine bases, 4 bases, guanine, adenine, thymine, and cytosine. These bases are placed in a unique order to code for all of gene and all microorganism or living organism. This figure shows the structure of DNA. The DNA consists of 2 strands, one 5-3- and the other strand is 3-5-. Each strand consists of nucleotide. Each nucleotide consists of phosphate, sugar, and base. 2 types of bond in the DNA molecules. Hydrogen bond, which bind one strand with the other strand through bases with 2 types of hydrogen bond. Adenine and thymine, 2 hydrogen bond, and guanine, cytosine, 3 hydrogen bond. While within this strand there is another bond called phosphodiester bond, which link one nucleotide with the other nucleotide in the same strand. What is the physical and chemical properties of DNA? The DNA is a long polymer made from repeating unit of nucleotide. These 2 strands entwine like veins as a double helix. The nucleotide repeats contain both the segment of backbone of molecule which hold the chain together and a base which interact with the other DNA strand in the helix. In general, a base linked to sugar is called nucleoside. So what is the difference between nucleoside and the nucleotide? If the base is linked to sugar only, it is called the nucleoside, while base linked to sugar and phosphate is called nucleotide. If multiple nucleotides are linked together as DNA, this polymer is referred as polynucleotide. So the DNA is called polynucleotide. Why? Because the DNA, it consists of many nucleotides connected with each other, so it is called polynucleotide. So should remember what is the difference between nucleoside and nucleotide. The DNA double helix is stabilized by hydrogen bond between the bases as mentioned before, attached to 2 strands. The 4 bases found in DNA: adenine, cytosine, guanine, and thymine. The 4 bases are linked. The 4 bases are shown below and are attached to sugar phosphate from the complete type. The bases are classified into 2 types: purine bases and pyrimidine bases. Purine bases divided in adenine, guanine. Pyrimidine bases, cytosine and thymine. There is a 5th base called uracil in RNA molecules, okay, instead of thymine in DNA. What is base pairing rule? The base pairing rule means each base is complementary to the other bases, meaning adenine should combine with the thymine and guanine combine with cytosine. As base pairing rule, GC base pair with the 3 hydrogen bond and the AT with 2 hydrogen bond. This is called base pairing rule. Two important factors. What is the factor determining the strength of the association of DNA? Number 1, GC content. Number 2, the overall length of DNA. How can measure this strength? By determining in the laboratory, the strength of the interaction can be measured by finding the temperature required to break the hydrogen bond. This is called melting temperature. Melting temperature is the temperature required to break the hydrogen bond. So, the melting temperature is directly proportional to the strength of DNA. بمعنى ان كل ما الميلتنج تمبريتشر تبقى higher معناها ان ال DNA strands اقوى، فبتتناسب طردي مع ال DNA لان محتاجة تمبريتشر اكتر عشان اعمل بريك للهيدروجين بوند. معناها ان في GC كونتنت اكتر، معناها ان ال DNA strength is higher. بتاع ال DNA. This sequence to show what is the mean by sense strand or coding strand, antisense strand or template. If any sequence of bases A, T, G, G, A, any sequence, this is called coding strand or coding sense strand from 5' to 3'. The complementary strand is the 3' 5'. A is complementary to T, T is complementary to A, G is complementary to C. If you need to make messenger RNA from this template, this messenger RNA T هيقابلها طبعا ال A هيقابلها اليو لأن أنا بعمل messenger RNA. اوكي، ده مش للحفظ، ده مجرد لو في سيكونس واقول لك مثلا قول لي السنس ستراند ايه او الانسنس ستراند ايه او how to make mRNA from this template. اوكي، طيب في definition صغير اللي هو super coiling. What is the super coiling? Super coiling mean the DNA can be twisted like a rope in a process called DNA supercoiling, and this supercoiling measure how the total length of DNA or the large lengths of DNA are compact inside the nucleus in a small volume. This is called supercoiling. Two types of supercoiling, positive and supercoiling. What is the biological function of DNA? DNA usually has a linear chromosome in eukaryote and circular chromosome in prokaryote. The set of chromosomes in a cell make a genome and information carried by the DNA in the sequence called a gene. A genome structure located in cell nucleus in eukaryote and mitochondria, chloroplast in prokaryote. The DNA is held with a regular shaped body in cytoplasm called nucleoid structure. In many species, a small fraction of the total sequence of genome encode protein. For example, 1.5% of human genome consists of protein coding or gene or called exon, with over 50% of human DNA consisting of non-coding repetitive sequence, and this repetitive sequence encode functional non-coding RNA molecule which are involved in regulation of gene expression. بمعنى الجينوم كله نسبة صغيرة منه هي اللي تبقى expressed أو functional. يعني إيه functional؟ أو بنسميها الإكزون جين. يعني إيه function؟ يعني يحصل لها expression وتديني functional protein. فمن التوتال جينوم بيبقى نسبة بسيطة بس هي اللي بتبقى functional جين أو بنسميهم الإكزون. أما بقية الجينوم بيبقى عبارة عن repetitive sequence أو حاجة بنسميها انترون أو non-coding sequence بنسميها انترون بيساعد على regulation of gene expression or الإكزون. طيب هنتكلم دلوقتي على DNA replication. قبل ما نتكلم على before start DNA replication, we mention some enzyme which is important in DNA replication. Firstly, nucleases and ligases. What is the nuclease enzyme? Nuclease enzyme, this definition should remember. Nucleases are enzymes that cut DNA strand by catalyzing the hydrolysis of phosphodiester bond. Phosphodiester bond, which is the bond linked between nucleotide and the other nucleotide within the strand. Two types of nuclease enzyme, exonuclease and endonuclease. Exonuclease are nucleases hydrolyze a nucleotide from the end of DNA strand, while endonucleases cut within this. Another enzyme called the ligase enzyme. Ligase from ligation. Ligase are enzyme used in rejoin cut or broken DNA strand using the energy from ATP or NAD. Ligases are particularly important in lagging strand of DNA replication, as they join together the short segment of DNA produced at the replication fork into a complete copy of DNA. Another enzyme called helicases enzyme. Helicases enzyme are protein that break the hydrogen bond between the bases and unwind the DNA double helix into a single strand. And this is important in replication because the double strand should be break to give a single strand. This should be, or the enzyme responsible for this is the helicase enzyme. Another enzyme called polymerase enzyme. Polymerase enzyme, this enzyme synthesizes a polynucleotide chain from nucleoside triphosphate. They function by adding a nucleotide into 3-hydroxyl group of the previous nucleotide in the DNA strand. All polymerases work in 5' to 3' direction. Polymerases are classified according to a type of template that they use. In DNA replication, there is an enzyme called DNA-dependent DNA polymerase. Make a copy of DNA. The DNA replication is accurate process because it is the vital process for the cell. The accuracy of the DNA replication is due to, first, proofreading activity. The polymerase recognize the occasionally mistake in the synthesis reaction by lacking of base pairing between mismatched nucleotide. If mismatched is detected, a 3' to 5' exonuclease activity is activated and the incorrect base removed. And this made by proofreading enzyme or polymerase with proofreading activity or with the exonuclease activity. If any mismatch between the nucleotide, the exonuclease activity or proofreading enzyme from 3' to 5' remove the mistake base and or incorrect base and put the correct one. DNA replication make a copy of DNA. The DNA replication begins with a partial unwind of the double helix at an area known as replication fork. This unwinding is accomplished by enzyme called, as mentioned, DNA helicase. This unwind section appears as under electron microscope as a bubble known as replication bubble. This is the copy of this is the DNA with the black color with the original strand, and there is a yellow color which is the primer, and new strand is red color. The double stranded DNA start from 5' to 3'. This is called leading strand, and the other strand is 3' to 5' is called lagging strand. The primer, which is yellow color, is complementary with the one strand, and the strand is completed as a continuous strand, so it's called leading strand. In other side, two primer in the lagging strand complementary with the lagging and give two part or Okazaki fragment which is linked with the ligase enzyme. So one copy of the DNA will give two copy, one old and other is new. Okay, this is the replication. One copy give two give 4 and so on. The DNA replication is conservative or semi-conservative. What is the equation? The DNA replication is conservative or semi-conservative. The DNA replication is called semi-conservative process. Why? Because each new strand is complementary to its old template strand. The two identical new copies of the DNA double helix are produced during replication. In each new helix, one strand is the old and the other strand is synthesized as a result, describing by saying that the process is called semi-conservative process. This is the diagram for the process is conservative or semi-conservative. The original strand is green and the new strand is red. The DNA after replication, one strand is the old and one strand is the new. So one is the green and the other is red. So it is semi-conservative process, not conservative process. Okay, this finished DNA and the enzyme used in DNA replication. Okay, we start now with the protein synthesis. So before start with the protein synthesis, should mention types of RNA used in protein synthesis. What is the types of RNA? Two types of RNA very important: messenger RNA and transfer RNA. Messenger RNA, message, record, معنى record or message. This nucleic acid that record the information from DNA in the cell nucleus and carry into the ribosome called messenger RNA. So messenger اللي هو بيعمل record للمسج. transfer RNA الفانكشن بتاع transfer RNA is to deliver or to bring amino acid one by one to protein chain growing at the apparatus used for synthesis of protein called ribosome. The special apparatus for making protein called ribosome. Ribosomes are made of two subunits, small subunit and a large subunit. And the difference between ribosome in prokaryote and eukaryote mentioned before, ribosome in prokaryote is 70S, in eukaryote 80S. 70S consists of two subunits, 30S small subunit and 50S large subunit. While in eukaryote, 80S, 40S small subunit and 60S large subunit. These two subunits open up like a Pac-Man, allowing the messenger RNA message to slide through. Once the messenger RNA is in place, the protein synthesis is ready to begin, and the two subunits close again so that the mRNA is now between the two subunits. The next player in the protein synthesis is the transfer RNA. This molecule is responsible for bringing the proper amino acid. Remember, the messenger RNA is now held within the two subunits of the ribosome and is relatively immobile, and the amino acid, the building block of protein, are floating free in the cytoplasm. Bringing amino acid down to messenger RNA, this by the action of tRNA. So the tRNA molecule acts as a taxi or delivery, whose job is to read the code from mRNA and bring the corresponding amino acid into place. So tRNA called a delivery or a taxi because read the message and bring the corresponding amino acid in place or proper place in ribosome. What mean by corresponding amino acid? Corresponding amino acid mean Each tRNA molecule has two own set on 3 bases which you called anticodon. This anticodon is complementary to mRNA code. The other end of tRNA molecule has an acceptor site where tRNA specific amino acid will bind. Mean the corresponding amino acid which codon is complementary to anticodon. So the mRNA has a code. The anticode of tRNA is complementary to this code is called corresponding amino. 3 definition is important. Triplet, codon, anticodon. Triplet mean 3 bases on DNA. Codon mean 3 bases on mRNA. Anticodon 3 bases on tRNA. So 3 definition: triplet, codon, and start now with the protein synthesis. The process of protein synthesis occurs in 3 steps: initiation, elongation, and termination. Initiation mean to form a initiation complex. Protein synthesis is initiated when mRNA, ribosome, and the first tRNA molecule carrying its methionine amino acid come together, and this is called initiation complex. When initiation complex is formed, the protein synthesis is initiated. The ribosome is inactive when it exists as 2 subunits, the large one and the small one, before it contact with an mRNA. The small unit of the ribosome will initiate the process of translation when it encounter with an mRNA in the cytoplasm. So, when the ribosome present small subunit apart from large subunit, it is inactive. No protein synthesis is formed. But when small subunit come in contact with the large subunit, and between them mRNA and the tRNA come from the cytoplasm inside the ribosome, it is called active ribosome, and the initiation complex is formed for the proteins. This figure shows the ribosome is inactive because small subunit away from large subunit and the messenger RNA away from the 2 subunit, and the tRNA in the cytoplasm and C above. This no protein synthesis started because the ribosome in this case is called inactive. In this figure, the initiation complex is formed and the protein synthesis is initiated. Why? This is small subunit and this is the large subunit. Between them, close between them, messenger RNA and start codon AUG, which is the start codon in messenger RNA, is complementary with the UAC of tRNA carrying methionine, and this is the initiation complex. And when the ribosome in this form, it is called active form of ribosome, and the protein synthesis is initiated. The first AUG codon in the 5' end of the mRNA act as a start signal for the translation of amino acid called methionine. So this codon and amino acid will always be the first in any and all mRNA molecule. So any mRNA molecule or any protein is started with the methionine because AUG is the start codon for this amino acid. The initiation is completed as mentioned when methionine tRNA occupies one of the 2 binding sites of the ribosome. Since the first site is the site where the growing peptide or protein or P site is released, and there is another side to the 3' direction of P site is called A site or attached site, where the incoming tRNA will attach itself. The second step is called elongation step. The incoming tRNA will bind to A site next to tRNA carrying the methionine on the P site. All available tRNA will approach the site and try to attach, but the only tRNA which will successfully attach is the one whose anticodon is complementary to the codon of A site on mRNA. As mentioned before, what is the complementary, or what is the complementary mean the AUG is in messenger RNA carry the anticodon for AUG, which is the methionine, and second amino acid which codon any code the complementary on tRNA who is attached to this code to give the second amino acid and so on until the message is finished. So in elongation step, each amino acid carrying, or each tRNA carrying amino acid will enter the ribosome, and the one which is successfully attached is complementary to the codon of the A site on RNA. Let us say, for example, the second tRNA that land next to methionine tRNA is leucine amino acid. These two tRNA holding methionine on P site and leucine on the A site are now next to each other. What happen exist is critical for the building of. In order for protein chain to form, amino acid must be attached, linked together, and this link between amino acid is called peptide bond. The amino acid continue to be linked until the protein is finished, and this special type of bond is formed by enzyme called peptidase enzyme. So the peptidase enzyme forms the peptide bond. What is the function of peptide bond? Link the amino acids with each other to form protein molecules. Once the bond has formed between the two amino acids, the tRNA on the P side leave and pass its amino acid on the tRNA on the A side. The last step is called termination. The elongation procedure continues until the proper protein is completed, until stop codon. What is mean by stop codon? 3 code UAA, UGA, or UAG signal the end of the process, because there is no tRNA that is complementary to the stop codon, so the process of building of protein is stopped. Because this is called stop codon. Why? Because no any amino acids for this code, so it is called stop codon. An enzyme called releasing factor free the new made polypeptide chain or protein from the last tRNA, and the mRNA molecule is released from the ribosome, and small and large subunit fall apart.